Have you ever wondered how humans and chimpanzees have 99% identity across their DNA? Or how researchers can identify tiny mutations in a virus which is hopping across the globe during a pandemic? Well, the answer does not lie in a microscope. It lies in your very own computer. Welcome to the world of bioinformatics, where biology and computer science interact with each other. At the very heart of bioinformatics is sequence alignment. Think of sequence alignment as a master key which help us unfold the very code of life, that is the DNA molecules, the RNA molecules, or the protein sequences to unlock their very secrets. So by looking at sequence alignment, we can identify our shared ancestry, our evolutionary history, or even also we can identify a marker or a biomarker for a certain kind of disease. So if you are interested in how sequence alignment works or what is sequence alignment, you are on a right track. Welcome to Bioenigma Uncovered. So the very first question is, what is sequence alignment and why does it matter? Let's break it down in layman terms. Suppose there are two recipes which are intended towards preparing chocolate chip cookies. Both the recipes have a similar kind of ingredients, that is sugar, flour, and they have like eggs and butter. One recipe intends to use baking soda and the other one intends to use baking powder. How the two recipes stack up against each other? Well, then we have to align the ingredients side by side, that is sugar against sugar, flour against flour, and so on. So sequence alignment is very much similar to that, but herein instead of recipes we are comparing strings of letters of DNA molecules, RNA molecules, or protein molecules, and by looking at those strings we can compare sequences from different organisms. Now the question arises, why does it matter? Why should we take a pain or why should we bother about sequence alignment? For this, we need to look at certain kind of biomolecules. For instance, say the 16S ribosomal RNA gene or the protein, and we also have myoglobin, we can also have like insulin, we can also have like cytochrome C. All these proteins perform similar kind of functions across different organisms. What does that mean? It means that these organisms or different type of organism shared a common ancestry, and this is where sequence alignment comes into picture. So I hope you have now got a fair bit of idea why do we need to perform sequence alignment. Before proceeding further, we need to look at certain basic terms related to sequence alignment. The first term here is homologous sequences. What are homologous sequences? Homologous sequences are those when two or more sequences share to have enough resemblance or enough identity. Now, the question arises, is there any specific criteria to determine homology across different sequences? Of course. When two or more sequences or the sequences under comparison, they share more than like nearly 40% of identity or 40% of resemblance across them, then it is said that we are in a safe zone, or the sequences under comparison, they are said to be homologous. If the sequences under comparison, they have like less than 40% and more than 20% of identity or resemblance across them, then we are said to be in a twilight zone. What does that mean? Well, it means that we are in some kind of ambiguity and we need to take additional measures or testing before ascertaining whether the sequences are confirmed or established as homologous to each other. The sequences under comparison have less than 20% of identity across them, then it is said that we are in a midnight zone, and we cannot call in this zone, we cannot call the sequences as homologous to each other. So I hope now you have got a fair bit of idea about what are these basic terms. Let's move forward. We need to understand some terms like identity or similarity, which are considered different or treated as different when we compare DNA sequences or protein sequences. When we are comparing DNA sequences, then the term identity and similarity are analogous to each other. As you can see on your screen, when we are comparing these two DNA sequences and certain positions, the A matches with A, as you can see here. It is said that the two sequences are identical as well as similar to each other. However, when we are comparing a protein molecule or a protein sequence against other protein sequences, and at a certain position threonine is matching identically at a certain position with threonine molecules, then the sequences under comparison are said to be identical. However, at a certain position, as you can see here, when amino acids with with similar physiological properties, say for example glutamate as well as glutamine, which are polar and charged amino acids, they are aligning with each other at a similar position, then these type of sequences, they are said to be similar but not identical. I hope you have understood this meaning, similar and identical, which are different when we are comparing protein and DNA molecules. Now the question arises, what are we actually looking at when we are performing sequence alignment or comparing sequences against each other? Well, there are three fundamental notions or three fundamental concepts we are looking at. The first one is a match, the second one is a mismatch, and the third one is a gap at a certain positions across string of letters when we compare DNA sequences. As you can see here on your screen, there are two DNA sequences. In sequence 1, we have the letters A, T, C, G, and in sequence 2, here again we have the same sequence, that is A, T, C, and G. So you can see that the sequences are identical to each other. In these conditions, these sequences are said to be matching with each other at all the positions. However, in the second example, the string of letters reads in sequence 1 as A, T, G, C. In second sequence, it is reading as A, T, G, and G. So at this certain position, you can see clearly here, C is not matching with G. This is called as a mismatch. So what actually a gap is when we are comparing two or more sequences? As you can see here on your screen, that in sequence 1, there is a string of 4 letters, and in the sequence 1, there are only 3 letters. In the sequence 1, it is reading as A, T, C, G, and in the sequence 2, it is reading as A, T. There is no letter at position 3, and it is followed by the fourth letter. So clearly this is called as gap. So this gap is a root cause of evolution or mutation, and it has led to the development of various species on this planet. So this gap can occur during the course of evolution by insertion or deletion, which are combinedly called as indel. So when we have like an additional nucleotide is basically inserted at the gap, then it is called as insertion, and if it is like omitted, then it is called as deletion. So by studying or by reading the matches, mismatches, or the gaps, that is insertions as well as deletions, we can identify similarities or differences across the sequences. Let's move forward and understand the different flavors of alignment. Just like you don't use a hammer to fix a screw, you don't perform similar kind of sequence alignment methodologies when comparing different type of sequences. There are two major methods by which we can categorize sequence alignment. First is how the sequences are lining up against each other. In this categorization, there are two types of sequence alignment. The first one is pairwise sequence alignment and the second one is multiple sequence alignment or MSA. In case of pairwise sequence alignment, as the name is clearly indicating, we are comparing two sequences. In case of multiple sequence alignment, we are comparing more than two, that is 3, 4 or multiple sequences against each other. Clearly in multiple sequence alignment, we are getting a high degree of information in a very short span of time because we are comparing number of sequences simultaneously. The second method is the strategy we are using to compare the sequences against each other. Or the first category here in this in this case is global alignment, and the second one is local alignment. Global alignment tries to align the sequences from the very first residue to the very last residue. You can assume here it as like we are reading two books from the very first page or to the last page, and we are like comparing the books from page to page. Herein we clearly know each and every information about the two books, or when we translate literally to the sequences, it means that we clearly have each and every information about the sequences under. However, in case of local sequence alignment, we perform sequence alignment or sequence comparison at a very tiny fraction or at a very small section or a position of the sequences under comparison. One thing you must know here is that global alignment is performed when the sequences under consideration have similar lengths. Means if you have two sequences which are having like 100 residues in one sequences and the 100 residues in the other sequences, then we perform global alignment. However, if the sequences are different in terms of their length or in the number of residues, like in the first sequences we have only 100 residues and in the other sequence we are having like 1000 residues, then we perform local alignment. So clearly local sequence alignment or local alignment is performed when two sequences are under consideration, they are distantly related to each other. To conclude, let's circle back to where we started. We talked about humans and chimpanzees, how are they alike, what is bioinformatics, what is sequence alignment, why it is required. When we talked about the certain fundamental terms related to sequence alignment, we talked about gaps, matches and mismatches. In the next video, we are going to cover about the types of algorithms with a specific focus on dot matrix algorithm to understand how sequence alignment is carried out. If you like this our content, then please don't forget to hit the subscribe button as well as the notification icon, and I'll be posting the next video on the types of algorithms very soon. Keep watching and keep supporting Bioenigma Uncovered. Thank you so much for watching this video.